here we go with our entrance slip over our central dogma. Now remember, just reviewing, the central dogma is that DNA is transcribed into messenger RNA. So that's the first part of it. DNA is transcribed into mRNA, and then the mRNA is translated into the protein, right? The central dogma, DNA to RNA to protein. All right, that's important to remember, folks. If I would think, I would think that you are going to see that somehow on the end of course exam when you take the bio part of it. Okay? So, this thing right here, this thing right here, what are we starting with right here? DNA. And it is only half of the strand, right? Okay? It has already been split because in order to make RNA, the RNA has to duplicate off of the DNA. So it has already been split. So if I go here and I look TAA. What is TAA going to translate to? T goes to A. I'm going to use a different color. T goes to A. What does A go to? U. And the other A goes to U, right? Now, what am I constructing here? A specific one, messenger mRNA is what I am constructing here. That is mRNA. That's important, okay? Because I'm going to show you a little bit here later why it's so important. But if I'm looking at A, U, U, I look at my codon chart, A, U, U is isoleucine, correct? So this is isoleucine, right? Now, my next three bases, and remember, three bases make what we call three bases make a codon that then translates for an amino acid. So my next three letters are going to be U, A, C, right? And U, A, C gives me tyrosine. Sound right? So this is tyrosine. And I can tell you this is going to go batty on me because I'm going to run out of room and I need to have plenty of room when I'm writing stuff out. Okay? My next one is going to be C, A. I look at C, A, that's glutamine. CAA is then GLUTAMINE. Okay. My next one is going to be G, C, and U, right? Because remember, uracil replaces thiamine. So I look G, C, U is alanine. Next is going to be G, U, G. G, U, G is valine. Valine, if you want to say it that way, right? Next one, excuse me, is C, A, G. CAG. Glutamine, we've already had that too, right? So that brings up a point right there that we had two different codons that coded for the same amino acid, right? That coded for the same amino acid. Huh? We'll have another one, okay. This one is going to be what? AAG. AAG. AAG is lysine. Is that how you spell it? Lysine. S-I-N-E. Okay, so that one goes to lysine down here. And my final one connected is going to be G, C, oh, you can't read that, G, C, A, G, C, A, yes, he's right, alanine. So if you did this correctly, folks, you should have isoleucine, tyrosine, glutamine, alanine, valine, glutamine, lysine, alanine. You have now made a strand of 1, 2, 3, 4, 5, 6, 7, 8 amino acids, right? Now, how many bases did it take to get those 8 amino acids? Should be 24, right? Because what's 8 times 3? 24. Okay? So there should be 24 letters across there, if you would count them, because every 3 bases give us an amino acid, right? Now, here's the reason that I wanted to say something about this, because let's say now, I've got a new page here. Let's say that I show you that I have mRNA. And I'm just going to make up G, U, A, C, A, C. Now, if I'm given mRNA, do I need to get the other strand to that? If I'm given mRNA, what do I do next? I find the protein, right? Because mRNA translates into the protein. Could I go the other way and get the DNA? Yeah, let's do that. Let's do both of them. What's GUA? GUA GUA is valine, right? So that's my amino acid that goes with that, right? What's my amino acid that goes with CAG, or excuse me, CAC? Histidine. Right? Now, what's my DNA going to be? Okay. C, A, T. What about here? G, T, G. So do you see how we could build off of RNA too? So it's important to know whether I have DNA, whether I have RNA. How can you tell? Yeah, look for the U's and the T's, right? If there's T's in there, it's DNA. If there's U's in there, it's uracil, it's RNA, right? Are we there? We okay with that? Okay. Okay, folks, what we have here, okay, with our say it with DNA, this is just a practice in which we will be making and we will be doing some of these decoding with the DNA. Now, what this process has done is it had made the DNA codes to go with letter symbols. Okay? So what we're going to actually be doing is we're going to take this DNA and go to RNA. So let's do that with this first one right here. I can definitely see that I have DNA. How do I know that? Jonathan? Has T's in it instead of U's, right? So, as I translate this, I'm going to go G, C, A, G, G, U, G, A, A. Would we agree with that? So I have now taken this DNA, and what have I made? I have made RNA, correct? Now, when I'm looking at this RNA, I go through and I look at the codons that they give me. My first three are G, C, A. So if I go G, C, A, it gives me what letter? It gives me letter A, right? What's my next three letters? G, G, U. It gives me letter G. My third codon is GAA. Gives me E. So what did I just spell there? Age. Okay. Let's do the second one. Let's do the second one. We got to make the mRNA first. So it's G, C, U, A, G, A, U, G, right? Now, I have translated that or transcribed that. Now, when I translate that, GCU is an A. AGA is an R. AUG. So I have spelt what? Arm. What I want you to do, folks, is I want you to do 3 and 4 right now. 3 and 4 right now.